communication courses for over 7 years. Welcome, Karen.
I have to unmute. Thank you, Krista. Hi. Yeah, so that was a great introduction, and I look forward to sharing my screen now to get into this lunch and learn with y'all. And it's actually asking me a question. Hang on. Dun, dun, dun. Perfect. Okay. And as Krista mentioned, just if you have any questions as we go along, go ahead and add them to the chat. I'll try to monitor as I can throughout this presentation to keep track of any questions you may have along the way. And so as Krista mentioned, this is about effective scientific presentations. And my name is Karen Thiebes, and I'm the founder of Simplified Science Publishing. And it's really fun for me to spend this time to share a lot of the tips and tricks I've learned along the way and how I apply in my own company for my own clients. And first of all, knowing that this is an audience where you have all likely attended a scientific talk, you may be familiar with seeing this face in your audience. So this is someone who is bored, confused, or both. And the goal of today is to really do my best to share some of these strategies that will help prevent this face from being in your own audiences, whether that's in your poster presentations or your scientific talks with slides. And in order to share that, I'm going to start with some of my favorite scientific presentation tips in 3 main categories. So I'll start with some tips on how to engage your audience. I'll then get into how to present clear main points. And then I'll review some strategies on creating effective take-home messages. And this is really something where these 3 main core components I consider essential to creating an effective talk, where if you don't have ideas or plans for engaging your audience, presenting clear main points, or making a clear take-home message, you're probably not doing as effective as a job as you could be in sharing all the information, whether that's your research or other types of information to your audience, where they can walk away saying, great, I get what you were trying to tell me, and I'm excited about what you were trying to tell me. So let's go ahead and jump in and start with those tips for engaging your audience. My first tip is to use images, right? Images are worth 1,000 words, and it's one of those things that you can always add. Illustrations. Illustrations can go a very long way in simplifying complex information into a clear story that gives your audience the context and the grounding for things like scientific information that can be very dense and very complicated. And finally, I'm going to go over some ideas for including interactions, having jokes, getting your audience to pay attention in a way that involves an interaction of some kind. So first, let's start with images. When it comes to images, my 3 favorite databases are Pixabay, and that's shown here on the left, Pixabay, FreePic, and Google. And Pixabay, this is just showing an example, is one where these are royalty-free images, where if I put in the word biology, if I'm studying red blood cells, if I'm studying DNA, there are a fairly large amount of free images in this database. FreePic is another great one. And Google, as long as you're using proper filtering techniques, is another great way to get images. But read the fine print. This is something where, as you all know, copyright rules are king, and you need to jump into those tools and read the fine print. And the easiest way to avoid copyright issues is to even just use your own photos. And I am actually just going to pop out real quick here so that you can see an example from Pixabay. Check in the chat. Where Pixabay, this is one where I put in bored man at the intro of our talk. You can see that this gentleman is who I chose from Pixabay. And when I mean read the fine print, I really mean just before you press download, there's almost always something where you can check the content license, right? So free for use under the content license. Go ahead and get in there and understand, can I use it for free? Can I use it without attributing the author, right? The nice, gentle person, Geralt, who gave us access to this board photo. I call him Board Ben. And you can just really make sure that you have what you need in terms of use before putting it into your presentations. All right, so that's just a tip for how to include some of those images using copyright in the right way. And another tip that I mentioned were illustrations. Now, I know not everybody is an artist that can create their own illustrations, but there are databases where you can download illustrations and scientific illustrations. My website has some. FreePic is a great one, and Canva is another great one. And all of these, you do need to read the fine print as well on using illustrations and creating your own custom scientific illustrations. But it's once you get into it, it's not that hard to start to use some of these templates, such as these vector templates, where you can, if you have cells in a dish, if you have a method you want to share, you can use these templates to build a custom method illustration or a custom summary illustration that really helps tell your scientific story more clearly than just words on your screen or words on your poster. And most importantly, I would say my third tip for engaging your audience is interaction. So this is something where you can tell a story or have a joke that just might get a chuckle. When people laugh out the gate of a presentation, you already have them on your side. And another great way to do this is to give people a why, right? So nobody comes to a talk thinking that they don't have a reason for being there. They're there for a reason. It can be really a great way to just tell them why they're there, right? Is it because they're there to help you improve your scientific research, right? Is it a small lab committee meeting and you're trying to get feedback to push your project forward? You can say that at the very start of your talk. This goal of this talk is to help me make this research project better. It's for me to help find out X, Y, Z, or it's for me to help improve this certain treatment method. And so the why is another really effective way, especially in the scientific world, to connect with your audience, to get them thinking and reminding them that they came here for a reason, right? And at the start of my talk, we used Board Ben. Board Ben, I said, you're here because you want to avoid this face in your audience. And it's also important to note that using human faces, especially if you're doing your slide presentations, is another great way to get engagement, because we are just naturally drawn to looking and spending time looking at people's faces. So I've actually done a study where if I had just an image that was drawn, let's say just this comic image, you are probably spending up to 2 to 3 times more time of your visual time looking at this face than looking at this cartoon. And so again, if you're just trying to enhance engagement here and there, human faces can be a really interesting way to set the attention in a slightly different way. Now, I'm going to jump into my favorite tips for making clear main points. And when we're trying to make clear points, especially in the scientific industry, there is a tendency for both posters and slide presentations to write sentences that are extremely complex, right? So my tip here is, if possible, wherever possible, reduce every single phrase on your slide or title on your poster to one main point action, right? Something is either going to increase, decrease, stay the same, and remove all excess description. So I'm going to show you an example here on this slide. This next sentence was used in my PhD publication, and it's good for a manuscript, right? Scientific publications require a lot more dense detail for accuracy and clarity than a presentation where it's a poster or a talk. And so this sentence I'm showing you here is based on my PhD research, says microRNA 218 direct spinal cord target mRNAs identified using a risk trap screen. That's fine if I'm reading this independently and I have a lot of context of a publication, right? That's telling me there's targets for this molecule and this is how they were identified. But the attention span during a presentation is so much lower. So if I were to do this and use this exact same idea in a presentation title or poster title, I can change that to the verb and the subject. Identified microRNA 218 targets, and remove all unnecessary description. And I really encourage you to, if you're presenting scientific information, find ways to reduce the main point to something was identified, right? Something increased, decreased, stayed the same, and a subject. A verb and a subject without any excess description can go a long way in taking your audience with you in the understanding of a presentation versus, losing attention, because this is a lot to absorb, both word-wise on a screen and just as somebody's talking. They're paying attention to too many things. So simplify, simplify, simplify. And now I'm going to go through just another example, very similar, but it's nice, I think, to see some examples and to try this brainstorm technique for yourself. So here's another sentence coming directly from, what goes into a manuscript for my scientific publication. The brainstorm is this long phrase of identification of microRNAs upregulated by this thing, islet 1, LHX 3, during motor neuron differentiation. Do the same exercise for you. Put your brainstorm, just whatever comes out of your head for the main point, and then try to make one better, right? Simplify it. MicroRNAs are upregulated during motor neuron differentiation. Remove some of the detail. And even better, remove detail again, right? MicroRNAs upregulated in developing motor neurons. Right? Moving excess detail to get that main point clear. And again, that's not that this is wrong. This brainstorm one isn't wrong. It could be used for your publication language. But for a presentation, you need to simplify it to make it clear. Okay, now, my favorite way to make clear points is to use the idea of spoon feeding, right? So for scientific information, especially with slides, it's helpful if you use animation to pull things in. Now, it doesn't have to move across the screen. You can use, and I'll just escape here, one to check chat and one to check the animation. But I'm just going to zoom out to show you that spoon feeding your audience can literally be as simple as pulling up your animation panel in your presentation using appear or disappear, or just using slides consecutively to make the information appear one thing at a time. Partly because movement attracts attention, partly because nobody can absorb a fully filled out slide filled with text and data. So think about different ways about how you can spoon feed that information on every slide. Or if you're presenting a poster, think about how you're going to direct somebody to spoon feed them, the method, the main point, the conclusion, and have that really clear in your mind. What is going to make sense to an audience in these tiny bites instead of all at once? And another important thing here is in making clear points, when I'm looking at all of these spoons, I'm looking at the red one. So it's perfectly fine to use color to draw the attention of somebody's eye to whatever is the main point. Let's say you had a graph and these were all bar graphs and you wanted to highlight a difference. You could add a box, and it's not what you're going to use in your presentation for your publication, but you can add a box just by adding a shape, changing the colors. I'll make it red. I'll remove the color. No fill, pardon me. Box, red, change the width. Don't be afraid to add colorful boxes around your data that draw the attention to the main point and animate it just to make that really clear on a poster or your slides. Again, this may not be kosher in a full publication format, but when you're presenting, it's perfectly okay to point out to someone what they should be paying attention to, and then very clearly tell them why that's important. For part 3, the take-home message tip is to pause to regain audience attention before delivering your take-home message. I paused awkwardly before moving forward with this slide, and it is scientifically proven that all of you likely heard the pause in all of my talking at you, and just took a second to say, why is she not talking? Nothing's on the slide. What's going to happen next? So it can be a very effective tool where if you have a very important message at the end of your talk, or if you're going to deliver the big news that's your conclusion from your poster presentation, pause. It's a very effective method, and it's sometimes silly. It feels silly to do it, but it works. So consider doing that for your take-home messages. And if you're doing a slide presentation, it can also help to use a unique slide design or change the tone of your voice for the take-home message. Again, there should be something in your talk that you're hoping people will walk away from and say, ah, that was why I sat down. Oh, that was what was explained to me, or that's why this research was important. Whatever that key take-home message is, try to identify that and then highlight it in this way to make sure your audience is paying attention and taking that with them when they leave. And my final tip in the tip list is to practice, practice, practice. It can be very helpful to refine whatever it is you plan to say, or whatever methods you're using to say those clear points. It can be helpful to refine when you practice in front of an audience, practice in front of your cat, practice in front of your family, whatever it is, your friends. Run this through a few times, and that will often create a really effective talk that just gets refined in ways that you wouldn't be able to do it unless you put through a little practice round to get it refined and where you want it to be. And now I'm going to just pause for a second here. I'm going to check back in, see if we have any questions. I don't think we do, which is good, because I can keep on going. So this next bit is, I went through the tips, and now I want to go through my favorite way to apply these tips, which is truly to get out a pen, get out the paper, and do the old-fashioned make an outline. And so if you can think about all the little tips and tricks I talked about in those previous slides, an easy way to apply those is to just get your piece of paper, have your engage your audience section of the paper, present clear main points part of your paper, and the take-home message part of your paper. And I encourage you, for any talk, before you start putting your slides together, before you start crafting the details of the poster, write down those engagement ideas, right? Is there an illustration or an image, maybe my cat, right, that's going to grab attention? This is Greg, by the way, and you might see him in the background in my video. What is an idea that will get people, I'll click one back, engaged in your talk as you start? And I do think it's really important to do the engage your audience bit at the very front of your talk. You can use these tips throughout to have images and make interactions throughout your talk, but it's most important that you do this at the very beginning of your talk. And my favorite thing to do is to really make sure that whatever image or engagement idea, sure, the cat is cute and fun, but I think the most effective, especially with scientific information and scientific communication, is to make a connection to that shared purpose, right? Is there a relevance to a medical treatment or to fundamental science or to whatever shared purpose it is that you are going to have for this presentation? That is usually what's going to truly make, I think, the most engaged start to your talk. And it can be the funny and the cute, like Greg. I would say the next thing you do after you have your ideas written down on how you might engage your audience, what images you might try to find, present clear main points. My favorite thing to do for this is to set a timer for 5 minutes. 5 minutes is actually a really long time when you have it set out and you're sitting down to write an outline, but have that full 5 minutes set and write down all of the main points of your research and your presentation. And then after that 5 minutes is up, take the time to circle only the 3 to 5 main points that tell your scientific story. When most people are presenting dense information like scientific research, they're tending to present too many points, and they can be bundled, they can be cut. Really try to look at your own brainstorm list and condense it down to the biggest points that will make it really clear to your audience what you're trying to share with them. And again, you can have those long-winded sentences at first that are more technical because you're maybe more in your technical part of your brain, and then spend that time to then pick your 3 to 5 main points and then refine those so that it's just verb subject, right? Extra detail you can talk about in the talk, but in your titles of the slide, in your titles that you include on your poster, use that really condensed, clear main point format that reduces all description to just the bare bones so that people's eyes don't glaze over just because they're looking at too many things, right? This is a visual thing more than an information thing. So don't be afraid to remove the rest. It's okay to let some information go away to then really make what you're trying to get across clear. And finally, after you've done this exercise, I do recommend writing down the one idea that you want people to take action on or to remember after your talk. And that will, having this guiding light may even help you refine your main points, right? You may be able to say, I know the end goal of this talk now. I can refine my main points to lead up and really help make sense of that final take-home message. And I'm going to check in here before I move to the next slide, see if anyone has any questions. We don't, which is good. Well, I can keep going, so I am going to keep going. Now, I had the option here to end my talk with a thank you and some ideas for courses. If you are interested in free stuff, like the slides, I have free slides. But what I'm going to do next is actually go into even more tips and tricks on presentation slides. So I'm going to keep this open for questions. If anyone has any questions about the topic or anything else about scientific illustration and design. And I'm going to go into this example where I'm going to share even more tips and tricks, especially when it comes to presentation slides, how you can apply everything I just talked about with what I've already started talking about is a dissertation example, right? So many of you, I think, are in very technical fields, and you don't have to be. You can also just be, in not a highly technical field, but in the field of science. This will apply no matter what your topic is in science. And so this PhD dissertation example is one I got published in Nature Communications. And what I'm going to do is show you an example PhD dissertation slide development that follows all of the rules I just talked about. Okay, so for this next set, if I'm making my own dissertation outline example, I'm going to make a connection to a shared purpose, and I'm going to make a slide that shows that my research can lead to potential therapies, and I'm going to use illustrations to improve their understanding. I'm going to only present 3 to 5 main points, and 3 points is all I need with very simple, this thing is expressed, this thing was down-regulating targets, and this thing is important for motor neuron development. And finally, I have a take-home message and a take-home slide example showing that microRNA tweaking is essential for motor neuron development and does the thing that I set out to prove. So let's go ahead and get started. So an intro slide, and I don't need the animation for this. An intro slide is always helpful. You don't have to have a bunch of crazy pretty images. It can be fairly simple. Just keep it clean and try to, again, limit how much jargon gets put on this main slide. The next slide is that why. Go ahead and ask why study XYZ, and then tell them why studying this thing is important. Studying motor neurons is important because they're required for movement, breathing, swallowing, and the loss of motor neurons often results in disease or death. That's an important why. And by uncovering the mechanisms that I talk about, you can bring researchers another step closer to defining treatments. Right, so just using 2 free source images, saying why am I studying this thing? Why should you care about this presentation? And answering that in a pretty simple format without being too creative can be a really effective way for scientists to have that initial engagement slide and not be too silly. Illustrations, right? So using illustrations to make it really clear when you're talking about the background of something can be really helpful. You can draw these, you can find these online to the best of your ability, and use animations. Right? There's 3 spinal cord cell types that were a part of my research. I label them. I animate, just bringing in text and shapes in groups. And that is spoon feeding the information and using illustrations to engage the audience and keep it really simple. Now, make it even more clear. Motor neurons are the ones that attach to muscle. You can be a little cheesy. It doesn't have to be perfect, right? This is not the most, this is a copyright-free image. These are my own little illustrations, but it doesn't have to be, you can get creative with how you embed these images and where you use these images with the animations to tell your scientific story. It doesn't have to be all seamlessly illustrated in the same way. And the next slides, they're just more of this, right? I'm just showing you that you can use animations, use only usually 2 main concepts on a slide at a time. And this is the next one I want to talk about. When it comes to animations, obviously you can use just this one. All that I was using for these were appear. Right, so if I go to PowerPoint, appear and disappear, and see the animation pane. These are just 3 groups set to appear in click order, on a click. A little more complicated way to do this is to hide things under different layers. So for this one, I'm just going to quickly do the animation where DNA makes mRNA, which makes proteins, microRNA is made, and it represses the expression of protein, right? It chops up this mRNA. In order for this animation to work, it's kind of crude, but it works pretty well. You can use shapes that are just hiding some of the things that move across the screen and appear in the right order. So this one is definitely a little more complicated than the appear, disappear. But if you have a complex story to tell and you're trying to say something foundational about your work, using layers where you can arrange them front and back and just organizing this in a way where you're hiding some of the information under things can kind of be a really cool method of adding and sharing mechanisms when it comes to your scientific research. And this is not that crazy, right? This is just a bunch of shapes that I've grouped. This is something that I have as a template that I use, but everything else is just a PowerPoint shape. PowerPoint can go a really long way in illustrating core scientific concepts if you're just a little creative about it. So that's just a tip that with animation and spoon feeding, you can get a little more creative and you can use selectively. You don't want to have things spinning in and out, but you can use this animation path to draw straight lines. I don't recommend the spiral, right? Keep it simple. To draw straight lines and have this start point and end point picked out for different parts of your slides. Okay, now this bit I do want to talk about. When I mentioned having 3 to 5 main points, if you're doing presentation slides, it is very helpful to provide your audience with an actual talk outline slide. So for this, right, I'm going to, and I'm not going to go into the details. You don't need to know the details, but my main point here for presentations is that having a bullet point list, letting people know what to expect for the talk, is something that people find really satisfying in an audience, because they've been told, here's the story I'm about to be told. And especially with scientific stories that are complex, it helps to have that map in front of you, right? So tell them in a talk outline, and then let them know what we're going to talk about next. So using a bullet point talk outline, it seems some people think, I don't need an outline. I strongly recommend always including it. It's a guidepost for your audience as you change complicated topics to move to the next topic. Now let's get into some data slides. My most favorite way to do data slides, like I said, using the spoon feeding, using the engagement tactics, is to start with a model on the left and data on the right, with that very simple, as simplified as possible, title at the top, right? So the model of a developing spinal cord and the results that show the data. This, keeping it to just a 1-2, is a great way when you have data on a slide to break up your data and not try to put a whole figure with all the panels on one slide. I think that's often a mistake people make, is to try to fit too much data on a slide, break it apart, and do the same thing. So this is the same thing. I'm showing a model on the left and the data on the right. A model with some words, a little more words on the left, a little animation to make it clear, and the data on the right. So again, this is just one sort of method, especially with data dense talks, that can help ground the information in ways that are digestible. And each time you go to the next main point, make that clear. Bring back that talk outline, highlight whatever's next in your talk. And in this case, I do have data and then model on the right. But either way, it's, again, I'm not over placing graphs so that people's eyes are trying to follow too many things all at once. Same thing. This is data that's related to each other, but it's not too much data all at once. And I could go like this forever. We're not going to get in there. So there's lots of different ways to do this. The next thing I really wanted to get across for tips, because this is pretty much all the same, of just data on the left and the right. Have that slide that stands out for the main point, right? So, and keep that main point simple. MicroRNA 218 is important for motor neuron development is a pretty broad scientific statement. And some might argue, especially if you're maybe defending your thesis, people would say, can you give me a little more specificity than that? But you've already talked about the specificity throughout all of the talk in the data details. I would say don't be afraid to simplify these titles so that people can easily absorb the information, even if some people are a little bit judgmental about the lack of scientific jargon included. And again, having some sort of graphics where you're just using, again, you can even just use PowerPoint to make some shapes and some things clear, is a great way to make your main points clear. And never forget your thank you slides. Always good to have that. And I do want to just quickly point out that I did not do this PhD research all entirely on my own onesie twosies. Right, there are some important people in my lab, especially those who are starred, who added some method and some data that were very essential to this talk and to this data that I showed you today. So always make sure that you include that in any presentation. Acknowledgements go a long way in both posters and your oral presentations to make sure you're giving credit where credit is due. And that is all I have today, Krista. So I don't know if you want to pause and see if anyone has any more questions, or if...
Oh, yeah, that was a very insightful presentation. I know I will be using a lot of that stuff in some of my own upcoming presentations as I present at conferences and as I get kind of ready to defend my dissertation. So this was awesome. Thank you so much. So let's see. I guess we can go ahead and end the recording, Giselle, and then kind of see if anyone would have any questions they would kind of like to ask in a more offline confidential setting. So feel free